Hey everyone, welcome to the channel. In today's video, we're going to explore something truly fascinating and insightful. I'll walk you through it step by step in a simple and clear way, so make sure you stay with me until the end. If you're new here, don't forget to subscribe and join the journey. Now, without further ado, let's dive into the science.
For decades now, the silicon chip has been king. It's the engine behind, well, everything. But what if the next giant leap in computing isn't built in a sterile factory, but grown in a lab? Today, we're diving into a totally new kind of computer, one that's literally built from the molecule of life itself, DNA. So, here's our roadmap. First, we'll look at why we're even pushing past silicon. Then, we'll explore why DNA is such a game-changing alternative. After that, we'll get into the nitty-gritty of how it all works with something called the strand displacement reaction. We'll see how that lets us build actual circuits, and finally, we'll geek out over what the future could look like.
All right, so let's start with the big question. Why bother? I mean, our computers are incredible, right? But what if they finally hit a wall?
Well, here's the deal. Our silicon-based computers are ridiculously power-hungry. You know Moore's Law, the idea that computers get twice as powerful every couple of years? It's kind of sputtering out. We just can't physically shrink transistors much more without things getting weird at the quantum level. And the gigantic data centers needed for AI, they're becoming a huge energy drain. We've basically hit what you could call the silicon ceiling.
This is where things get really interesting. The solution might not come from better engineering, but from biology. Instead of trying to build a brain out of silicon, what if we used the molecule that already stores the blueprint for all life? Let's start thinking about DNA not just as biological code, but as the ultimate computing hardware.
So why DNA? Well, for one thing, the information density is just insane. Get this, a single gram of DNA, that's about the weight of a paperclip, could theoretically store an entire exabyte of data. An exabyte is a billion gigabytes. We're talking about all the data from the entire internet just a few years ago fitting in the palm of your hand. It's mind-boggling.
So how on earth is that even possible? Well, it all comes down to the language they speak. Your computer, it's bilingual. It speaks in ones and zeros. That's it. It's a binary system. But DNA? DNA is fluent in a four-letter alphabet: A, T, C, and G. Right off the bat, you have a much richer, denser way to pack in information. Okay, so storing data is one thing. But how do you actually compute with it? How do you get it to think? The answer is this really elegant, almost beautiful process called DNA strand displacement, or DSD. This is the absolute core of it all, the engine that makes this entire thing go.
So, at its heart, DSD is just pure physics. You don't need a bunch of other complicated biological machinery. Imagine a single strand of DNA, let's call it the invader, comes across a pair of strands that are already bound together. If that invader is a better match for one of the strands, it just latches on and literally kicks the original strand out. A predictable, programmable reaction.
Let's break it down into 3 easy steps. First, initiation. The input strand docks onto a little exposed piece of the complex called a toehold. Think of it as the start here sign. Next, migration. The input strand starts zipping up with its partner, displacing the old strand one base at a time. And finally, release. The original strand is completely booted out, and now it's floating free. And that free-floating strand, that's your output. An input just created an output.
What's so amazing is that each piece of this molecular puzzle has a job that we can recognize from computer science. That invader strand, that's your input signal. The strand that gets kicked out, that's your output signal. The complex itself is basically the logic gate, and that little toehold, that's the on switch that gets the whole thing started. It's a perfect little machine.
All right, so once you can reliably control one simple input-output reaction, you can start snapping them together like Lego bricks. This is how we take that one tiny reaction and start building the fundamental components of any computer. We're talking about logic gates. And that's the key takeaway right there. That simple physical process is a logic gate. By carefully designing the specific sequence of the DNA, the A's, T's, C's, and G's, we can actually program these molecules to make decisions. It's a fundamental shift from etching logic into silicon to encoding it in a molecule.
And check out how elegantly this works. If you want an AND gate, you design the system so an output strand is released only if input A and input B are both present. For an OR gate, either input A or input B can trigger the reaction. And for a NOT gate,
The output is always on by default until a specific input comes along and displaces it, turning the signal off.
And just like with regular electronics, once you have your basic logic gates, you can start connecting them. The output from one gate, that little strand that got released, can become the input for the next gate in the chain. You can create these incredible cascades of reactions, building complex, integrated circuits that can perform some really sophisticated calculations. All of this happening in a test tube.
Okay, this is all fascinating science, but what's the payoff? What can we actually do with computers made out of DNA? Let's talk about the incredible potential here, because this is where it really gets exciting.
The applications are nothing short of revolutionary. Imagine tiny DNA nanorobots that patrol your body. They could use an AND gate to ask, is this a cancer cell? And is it this specific type of cancer cell? Only when the answer to both is yes does it release its drug cargo. We're also talking about super advanced biosensors for instant medical diagnoses, creating disease models in a dish to test drugs, and even building neural networks for AI with molecules instead of silicon, which would use a tiny fraction of the energy.
So this all brings us to a final, really profound thought. For all of human history, we have manufactured our tools. We've built them, etched them, assembled them. But DNA computing points to a future where our most advanced computers aren't built at all. They're grown. We're truly beginning to program life itself, blurring the line between machine and biology. The question isn't just how fast our computers will get, but what exactly they will be made of.